To begin with, to move on, I think that we ought to lay out some crucial terminology and concepts. And the first thing that comes to mind, maybe the most important for this field, is the distinction between quantitative and molecular genetics and where or from who they came from.
Right, good. Yeah, that's a good one. Probably even more fundamental, though, is the focus that, of our 3 billion base pairs of DNA, in the double helix of DNA. I'm sure everybody kind of knows that about the double helix. There are 3 billion steps in that spiral staircase of DNA, and that's what we call DNA bases. And we're identical. We have identical sequence for over 99% of those 3 billion base pairs of DNA. And that's what makes us human. That's what makes us walk on 2 feet and be natural language users. We call that a nomothetic or species-wide point of view, which is kind of an evolutionary perspective in some ways, talking about universals, say for the human species. And in terms of development, children develop on average, they say their first word at 12 months and 2-word sentences at 18 months. Those are nomothetic, universal perspectives. And that's what the 99% of the DNA is about. But the 1% that differs allows us to ask, to what extent does DNA differences, inherited DNA differences, make us different in behavior? And in my sort of genetics, we're talking about individual differences. Why do we differ? Why are some of us heavier than others? Why are some people better at school than others? Why do some people have schizophrenia? So it's a really important distinction because there's no necessary relationship between these different levels of explanation. That is at the species-wide level versus the individual differences level. So that's probably even more fundamental. Now, the quantitative genetics, behavioral genetics is just the genetic study of behavior, but we're talking about individual differences. And that includes molecular genetics, DNA studies. That's the hot thing now. But it also includes quantitative genetic studies for about a century. These are like twin and adoption studies. They're quasi-experimental designs that allow us to ask the extent to which familial resemblance, things run in families, to what extent is that due to nature or nurture? So that's what the twin study and the adoption study are about, and we can talk about those. But in terms of this bigger picture then, behavioral genetics is the genetic study of behavior. That includes both molecular genetic, that is DNA studies, but also these older quantitative genetic methods that get at the bottom line of genetic and environmental influence. And they're really just as much about the environment as they are about genetics, because our environmentalistic studies for the last century in psychology are often confounded because they don't even think about genetics. So they show parents do something, or that there's a relationship between how heavy parents are and how heavy their kids are, whether they're schizophrenic or not. It runs in families, and it was just always assumed it runs in families for reasons of nurture, ignoring the fact that parents and offspring are 50% similar genetically. So it could be, and it turns out it is, largely genetic, that is familial resemblance. So the excitement in behavioral genetics now is that these 2 worlds of genetics, molecular genetics and quantitative genetics, are coming together in what I call behavioral genomics. The genome is okay. And they have their origins in the late 1800s. I think that's what you were getting at. So early on, these 2 worlds were completely divergent. The molecular genetics, the origins, was Mendel studying single gene characteristics and understanding the laws of heredity. And the goal there was to understand how genes work. In contrast, Galton, who was the cousin of Darwin in the 1870s, initiated the field of what we call quantitative genetics, which focuses on traits, phenotypes we call them. So behavioral traits, or a lot of the early work was with plants and animal husbandry. And a lot of that dealt with breeding, for example. For tens of thousands of years, humans have known that you can breed for traits. And although they didn't have any formal idea of how inheritance works, they kind of got the idea it has something to do with inheritance. The fact that you could select parents who would then have offspring that had characteristics you liked if you did that for several generations. So these two worlds of genetics were very different because the Mendelians, as they were called in the early 1900s, were focused on single gene disorders. And that's what a lot of molecular genetics was about. And by a single gene disorder, I mean a DNA variant, a mutation that is necessary and sufficient for the trait. So Mendel studied 7 of those in the pea plant. One mutation, for example, caused the seeds of the pea plant to be wrinkled. And it's necessary and sufficient that a pea plant would only have wrinkled seeds if they had that mutation. And if they didn't have that mutation, they would not have wrinkled seed. They'd have the nice, smooth seeds that you expect to see in a proper pea plant. So when people rediscovered Mendel, he wrote in 1869, and nobody noticed him until 1900s, sort of 1903. Darwin, it turned out, had a copy of Mendel's paper, but it was uncut. In those old days, you had to actually cut the pages. They were all stuck together in a portfolio, and you had to slice them, and they weren't sliced, so he couldn't have read it because he'd have to peek in there. So if he had, though, it would have solved his major problem in the theory of evolution. Where does this variation come from? And the essence of Mendel and understanding mutations is it's random. Mutations are random, but what natural selection does is it takes that variation and channels it in directions that involve adaptive responses like survival of the fitness survival. You have more offspring, basically. So when people found out about that in the early 1900s, they started looking for single gene disorders. So they were looking for these Mendelian segregation ratios. If it's a single gene, I don't know if you want me to go into all this, but you get these 3-to-1 ratios of mutations. But the important thing is there's ways of looking for these single gene disorders, and they thought they were finding them. But they thought that's the way genetics works. If it's genetic, there's one gene and it causes this thing, the behavior you're looking at. Now, on the other side, the quantitative geneticists, they thought this was crazy because they've always been studying phenotypes in animals, in plants, in humans, and they know everything you study is normally distributed. They're not just either or, like Mendel's pea plants, where you either have smooth seeds or you have wrinkled seeds. They're dichotomous traits, which is the hallmark of a single gene disorder. But so they thought Mendel's whole laws of inheritance must be bonkers or just something specific to the pea plant, because they knew that all characteristics, important characteristics for animals and plants and humans, are normally distributed. I'm sure people know what the normal bell-shaped curve looks like. And so in 1918, the foundational paper of quantitative genetics was published by Sir Ronald Fisher that put these two together. And he just simply noted, it's a very opaque paper, 48-page paper that nobody actually reads because it's very hard to understand. But the message was simple. He's saying Mendel's laws of inheritance could be correct, but if a trait is influenced by several genes, it will quickly look like a normal distribution. And so he showed that very nicely and graphically, that all you need is 2 or 3 genes, each of which have 2 forms, 2 alleles, and with a little bit of environmental variation, it will create something indistinguishable from a normal distribution. And so that's quantitative genetics, polygenic, where there are multiple genes involved. But on the other hand, we now know that there are thousands of single gene disorders in humans. But the thing about them is they're very rare. Like some of the more
like
common
Huntington's.
ones, 1 in 10,000 are the most common ones. Many of them are 1 in a million, 1 in 500,000. And they're often devastating for the person who has it, but they're not contributing much to the population as a whole. So if you think about medical disorders like hypertension, diabetes, there's substantial genetic influence, but single genes don't really make much of a difference. The genetics comes from many, maybe thousands of DNA differences across the genome. And the important part of that, to get to the bottom line of this story, is that people learn about genetics from Mendel, and you say genetics, and they think single genes, determinism, hardwired. And those 10,000 single gene disorders that have been identified, they are hardwired and deterministic. I mean, if you have the mutation for Huntington's disease, which is a neurodegenerative disease that killed Woody Guthrie, for example, if you were Arlo Guthrie, his son, you'd have a 50% chance of inheriting that. It's a single gene necessary and sufficient for the development of Huntington's disease. And it will kill you unless something else kills you first. It doesn't matter what your diet is or exercise regimes, it will kill you. So the trouble is people learn about genetics from Mendel, but when you get into the traits like common medical disorders or quantitative traits that we study in psychology, it's not a matter of single genes. It's polygenic, and that switches from determinism to a probability approach, where it's probabilistic rather than predetermined programming. And it's such an important distinction. And so I'm glad you brought that up, because I know people aren't often interested in the history of it, but it is important to recognize that if you learn about genetics from Mendel, you're going to be thinking single genes, and you've got to make the jump from that hardwired determinism to this probabilistic influence. So we don't talk about innate or deterministic. These are influences, genetics. So anyway, I could go on and on, as you can hear, but I'll take a breath and give you a chance.